Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I'm with RTI International's Forensic Technology Center of Excellence. Today's webinar is entitled Validation and Tutorial of NOCIt for Determining the Number of Contributors. Our subject matter expert for today is Dr. Catherine Gergachuk. She is an associate professor and Henry Rogers Chair in the Department of Chemistry at Rutgers University in Camden, New Jersey. She received her Bachelor's of Science in Physical Science and then her Bachelor's of Education from the University of Windsor, her Master's degree in Forensic Science from the University of Alabama at Birmingham, and then her PhD in Chemistry from the University of Ottawa. Her laboratory for forensic technology and integration is a research laboratory focused on the development of systems and procedures to improve forensically relevant bioanalytical processes. She does currently serve as the Executive Secretary of NIST's Biology SAC and is a member of the Journal of Forensic Sciences editorial board. She's also an editorial board of Forensic Laboratory Needs Technical Working Group, an expert working group on human factors in DNA interpretation, the American Society of Forensic Sciences, National Society of Forensic Genetics, and the Center for Computational and Integrative Biology at Rutgers University. Just our quick disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without any further ado, I would like to hand the platform and microphone over to our subject matter expert and presenter of the day, Dr. Catherine Gergachuk. Dr. Gergachuk?
Okay, thank you very much. I'm going to go ahead and share my screen now. Okay. So I'm going to let Josh go ahead and let me know if something goes awry, but from what I could tell, I seem to be sharing the screen with everybody. So let me just begin and thank everyone for coming. There were some technical issues last week when we tried to present this work. So if you're coming back a second time, I just want to take this opportunity to thank you for coming back a second time. And I actually changed the title a little bit on my slide as I wanted the title to reflect a little bit more closely what the presentation is going to be about. So it's really about validating NOCIt's probability on the number of contributors. And then I'm also going to present some early work on how we in this laboratory can calculate what we call the full likelihood ratio across all number of contributors. The main part of this talk is going to be focused on how we went about validating NOCIt, which is a module in a larger software system that we call Valid DNA. And as a cheat sheet, I just want to point you to this paper that where most of these data are already presented and graphed. So for the purpose of this presentation, I would like to focus mainly on demonstrating to the audience that, again, what I'll call full likelihood ratios, they actually can be obtained by integrating across all the signal and using the a posteriori probability on the number of contributors into the likelihood ratio. And anytime you see the letter N, that's always going to refer to the number of contributors. So the main point, for the main part of the talk, I'm going to describe the number of contributor interpretation gaps just to review with everybody any previous studies. I'm not going to focus too much time. I'm not going to focus too much on this, but I do want to just present that there are some studies already in existence that explicate that the number of contributors actually can be an important numerical value that one ought to consider during their interpretation pipeline. And so that's why about 5 or 6 years ago, we started to work on answering this problem. And when I say we, I mean myself and my collaborators. The two long-run collaborators that I'm going to mention here are Ken Duffy from the National University of Ireland at Maynooth, and then another collaborator, Desmond Lunn, who also teaches and works here at Rutgers University. So really, all the work that I'm presenting here is a collaborative effort between the three of our groups. So in today's talk, I am going to talk a little bit about how we went about validating NOCIt, and in particular, what do we mean, or what do I mean when I say validated, this software module or this platform? Well, like in most tests, or most times when we try to validate, we are talking about trying to assess the precision, trying to get a good sense of the accuracy of the system, trying to then compare it to previous or currently used, sometimes what's known as acceptable or accepted methods. And then we also tested whether or not NOCIt's APP were robust across multiple sample qualities. And then we looked at the performance of NOCIt at the boundaries. So typically with validation, we always, at least in this group, in my group, we always try to establish predetermined criteria before we go ahead and validate. That is, we try to say to ourselves, well, let us go ahead, document and record what our minimum expectations are, be ahead and implement this software into our own our own interpretation pipeline. So it's a little bit difficult, of course, to do that when one is developing a new system, but we knew ahead of time that at the very least we wanted NOCIt to perform quote unquote better than the maximum allele count method. And so we spent a lot of time asking ourselves, well, what does the word better mean? And are there some minimum criteria that we ought to meet? And so those questions are going to be part of the presentation today. The other thing I do want to point out is the word accuracy, because of course for our purposes here, we did design the validation such that we had a very good idea that of the quote unquote true number of contributors or nominal number of contributors. So different people use different terminology to describe that that measurement. But bottom line is we wanted to make sure that we were using electropherograms wherein we were relatively confident, or more than relatively, we were quite confident that we would expect to see signal from every contributor. And so in terms of our experimental design, we had to make sure that samples that we used were ones that were of relatively high, signal to noise, if you will, or relatively high sensitivity. So I am going to go into a little bit as well to sort of provide you with the background and justify the samples that we used in order to actually, quote unquote, validate the software. Okay, so just as a quick reminder, what is MAC, or maximum, the maximum allele count method in order to assign the number of contributors? Well, as far as I understand it, or at least as far as we implemented it in our lab, what we did is we applied an analytical threshold, and then any peak that crossed that analytical threshold or that boundary was reported or recorded, and then we counted the number of peaks that were above that analytical threshold and that were not considered stutter. And then we divide by 2 and round up. And this is the minimum number of contributors that can explain the evidence, and the technique by which we do this, I'll refer to it as MAC or MAC. And I think that's a pretty typical terminology that's used in the field. So one of the, I don't want to call it a problem, but of course, one of the things to note is that this technique will provide, again, the minimum number of contributors that may explain the evidence, but it definitely would not provide the probable and or indeed the maximum. And so there's some room in this area of forensics where we could ask ourselves or develop techniques by which we can answer questions of, okay, well, what's the most probable number of contributors given the evidence? And of course, this might lead to downstream interpretation issues, because there are some papers out there that suggest for mixtures, especially for minor contributors, or if the propositions are anchored in different ways between the numerator and the denominator of the likelihood ratio, then this ultimately may indeed have some sort of effect on the downstream, the final likelihood ratio, the final value that's reported. And in forensics, of course, we're always trying to reach a point where we have standardization across the lab and more repeatability or reproducibility between laboratories and consistency between laboratories. So to us, this was an important area of study because we wanted to try to do two things. One, develop a method by which we can compute the probability of the number of contributors given the evidence rather than reporting the minimum number of contributors that explains the evidence. And then we also wanted to assess whether or not there was, what could we do with this probability value that we obtain. And that's, of course, an important question too, because then we're asking, well, how does this APP, this probability of the number of contributors given the evidence, how does that fit into the overall pipeline? Colloquially speaking, someone might ask, well, yeah, that's great, but how do I use it, Catherine? How do I use that number? And that's something that I'll discuss today as well. But I just wanted to point out that there are some publications already in existence to suggest that this number can matter, and it can matter in specific cases, and therefore there is justification for looking into this type of question. So the first thing that we did is we tried to see through simulation whether or not there's some significant percent, if you'll let me explain it, some significant percentage of samples that might be quote unquote misclassified if we use the maximum allele count method. So one of my former students, who actually now works at the center, the Forensic Technology Center of Excellence, Sarah Norsworthy, she did a very nice experiment where she simply simulated 750,000 profiles. So not the peak heights, just the profiles, just the alleles. But she also perturbed whether or not the alleles would drop out or not drop out or be detected. And so she looked at various numbers of scenarios, different dropout rates, different number of contributors, up to 5 or 6 contributors, I should say. And then she simply then counted the number of alleles that were produced, and is reporting here that, for example, of the 166,000 profiles that that would be assigned in a MAC of 2, 90% of them actually came from true two-person contributors. See that, at least for this experiment, for this set that we simulated, by the time we're seeing a maximum of 6, or a minimum, I should say, of 5 alleles at any one locus, there's quite a bit of underestimation that could potentially be going on. So this is something that we thought was quite interesting. And again, it doesn't mean to say that every sample that exhibits a minimum allele count of 5 means that there's a 40% chance that it's coming from a 12-person, or excuse me, a 32% chance that it's coming from 4 people. It's just to say that the potential is definitely there to misclassify if we're relying on a maximum allele count method. So these data, or this table, was produced by Sarah, and these were just simulated data. But then if we actually looked at the number of samples that exhibited the maximum number of detected alleles, which is down here, and the proportion of those samples that actually came from red, which is 2 people, 3 people is green, purple is 4 people, and orange is 5 people, we see that even for the proved it samples, which are electropherograms, they're real samples that were run in our laboratory and published online here at lifty.com, we see that there's a great number of them that where we would misassign a lower number of contributors if indeed we were using the MAC method. So again, it's not to say that that happens every single time. It's just to say that the potential is there to underestimate the number of contributors if we say to ourselves, well, we're going to take the MAC assignment as the number of contributors to that evidence. Okay, so again, that's the lead up to say, well, what do we do in situations where maybe we understand that the MAC assignment is not the type of procedure we want to use, or the type of assignment we want to use, and perhaps we would like to start looking at using peak heights and probabilistic models rather than counting methods in order to develop a means by which to provide a APP, or probability of the number of contributors given the evidence. And so this is where NOCIt comes in. So we developed a system wherein we could essentially provide the probability distribution here. Hopefully you can follow my mouse, which is right here. So the probability distribution on the number of contributors. And so we can look at this and say, if this was my electropherogram, so this is simply an example of a global filer electropherogram from a 1 to 2 degraded to undegraded 2-person mixture, and the total target mass was 0.5 nanograms. So it's interesting because here, I couldn't really show it, but there were some peaks near the baseline that suggested that there could be a third minor contributor. And so when we ran this sample through NOCIt, we obtained the following APP distribution. And the way to read this result is simply to compare to the y to the x and read it as, well, the chance, or the probability, that zero contributors given the evidence is essentially near 0, and the probability that 1 person contributed given this evidence is again essentially 0. And then what we see is that the chance, or the probability, of 2 explaining the evidence well shoots up to 0.74. But we also see that there's a non-zero probability, or non-negligible, I'll call it a non-negligible probability, that n explains the evidence quite well at 0.26. And so this is quite interesting to us because it suggests two things. One is that there's more than one number that might explain the evidence, and there's more than one number that explains the evidence quite well. So even though the highest probability is affiliated with the N of 2, it's not to say that 3 is negligible. And so the way we look at this result is to say that it suggests to us perhaps reasonable numbers of contributors to exclude from downstream interpretation. So unfortunately, I'm not going to have a lot of time to go into the biological models, but we did publish some of the developmental validation for the biological and analytical models, and it's available in the proceedings in 2018 from IEEE, and then it's also available at BMC Bioinformatics. So the BMC Bioinformatics paper is open source, so you all should be able to find it should you have interest in looking at the specifics of the models. So the other thing we wanted to point out, or I wanted to point out, is unlike the MAC method, which sort of relies on the application of an analytical threshold, and indeed to some degree it relies on the application of what we call a stutter filter, NOCIt doesn't rely on the application of an analytical threshold at all. And so this becomes quite powerful, at least we believe it becomes quite powerful, because what it means is that we don't have to, or we could develop a laboratory system wherein we can be relatively confident that we are observing signal as long as the copy, an amplifiable copy of DNA made it into the PCR tube. Okay, so in terms of the validation that we published in just recently in July 2020, I just want to spend some time talking about how we validated NOCIt. And we spent a lot of time running the 815, a total of 815 global filer samples. And we specifically chose samples that were amplified for 29 cycles and injected for 25 seconds on a 3500. So just to note, it's not a 3500 XL, it's a 4-cap 3500. The reason we decided, again, to use these samples in particular rather than some of the samples that were amplified using a lower PCR cycle number is because with these laboratory conditions, we are quite confident that we are seeing signal from each of the contributors. Okay, so what that means is when we compare back to what I'm going to call the true number of contributors, I'm relatively confident that the signal represents, at least to some degree, some portion of every contributor. So here we just to provide you with the numbers, we tried to disperse the number of samples as evenly as we could given the open database that is available. So we used 100 single source samples, 193 two-person samples, 173 person samples, etc. And the total template mass ranged anywhere from 0.5 to about 1 copy-ish worth of DNA for the single source samples, and 5 copies and so on for the up to 5 contributor samples. And again, the reason why it was engineered this way is to ensure that regardless of, or depending on the ratio mix, we were relatively sure that we would have signal from that, from every contributor represented. Okay, so down to the validation, the first thing that we did is we took a look at the SWGDAM guidelines for validating probabilistic systems, and notably there's no guide or SWGDAM guideline or OSAC guideline that expresses how one might go about validating a number of contributors software. So they specifically focus on the likelihood ratio application, but they being SWGDAM. So what we did is, knowing that we took the principles of that SWGDAM validation guideline and applied it to this validation and just followed along. And instead of, for example, every place we saw likelihood ratio in that document, we replaced it with APP on the number of contributors and tried to ensure that we were following those guidelines to the best of our ability, even though specifically guidelines to validating number of contributor software doesn't exist. At least to my knowledge, it doesn't exist. Okay, so one of the things that SWGDAM asks developers to do is to establish repeatability and make sure that whatever system we are using, that it's repeatable. And so we also wanted to make sure that if we changed any of the NOCIt settings that we believed could influence repeatability, that we did that we changed those, and then we of course we recorded the impact not only on the time it takes per sample, but on the final result. So let me go through this slowly. We, in essence, tested 3 different conditions. And here on this table, you will see that there are a number of inputs that could potentially impact NOCIt's repeatability. And we focused on modifying 3 of these. So here you'll see that what we call the multiplicative factor was changed. And you'll also see here that we changed what is the sample batch size. So if, for those of you who are interested in knowing the particularities of what each of these terms mean and how they impact the algorithm, I would invite you to go to the NOCIt validation paper, which explains exactly how each of these inputs affects the algorithm. So it's a little bit beyond the scope of this talk, unfortunately. But, nonetheless, we changed these and we then recorded the average runtime per sample, and we were using a 6-core hyper-threaded computer. So typically we were in the tens of minutes of ranges, minutes I should say, for each sample. Some samples obviously took longer to run, and some took as low as 10 minutes, and some samples took as much as 4 hours. One of the things I do want to point out is that NOCIt allows the user to define a runtime limit. And so this is in seconds, so this represents 4 hours. And so what happens after 4 hours, if this is set to 4 hours, is the run will stop prematurely, and it'll let you know that, it hasn't reached convergence within the 4 hours. And typically what we'll do is we'll just restart the run and then allow it to run a second time. And so this number can be set to any value, and it's user defined. So this is the results. These are histograms of basically what we're going to call the APP range. So if we imagine that we ran NOCIt using each of these conditions 3 times across the 815 samples, then what we're plotting here is the maximum difference between the APP for the most probable number across all three replicates. So that's a little bit convoluted, but let me show you an example. So if I ran one sample on NOCIt, and the first time I ran it, the maximum A posteriori probability, so what we'll call the map, instead of the MAC, we're going to say, okay, the most probable number of contributors was at n equals 2, and it was 0.9. And so in the next run, if the maximum probability was at n equals 2, but this time it was, at 0.7, and the third time it was at 0.8. What I'm plotting here is 0.9 minus 0.7 across all the 815 samples. So if this bin, which is between 0.9 and 1, means that one run had, a probability close to 1 for 1n, and in the second run, it would have NOCIt's output would have suggested that another number was most probable. So what that means is we really want most of our samples to be near 0 or have high repeatabilities or small APP ranges. And so here we'll see that for condition 3, for example, even though for each condition we had pretty good results, you start seeing this last increase significantly as we start decreasing the sample batch size and the multiplicative factors. So now it's choosing condition 1 or condition 2 as the method of choice, and we ended up choosing condition 1 simply because we were able to withstand an average of a 40-minute runtime for sample on our computer. And so this was, in our view, a reasonable runtime for all of our research samples. And it provided us with very good, what we would classify as very good reproducibility across the 3 runs. So along those same lines, we also started looking at what we'll call the accuracy. So what's the probability that the true number of contributors is greater than 0.001? So again, what does this mean exactly? So if you go back to this graph, this is the output of NOCIt. So if we as the laboratory individuals can establish a threshold by which we say, okay, if the APP is less than this value, if it's, put another way, if it's too close to 0, that probability for that number of contributors is so low, I'm not going to include it in my downstream interpretation processes. Conversely, we could say, well, we're not interested in running a range. We're just interested in choosing the highest, the number of contributors associated with the highest probability. And so the question that we were asking ourselves was, well, if it's true that one is in the lab where you only want to choose one N, is that going to impact downstream interpretation? And if so, by how much? So the bar chart I was showing you earlier was, and I'll get back to it in a second, was basically we said, okay, well, for accuracy purposes and for these preliminary experiments, we just want to make sure that NOCIt is providing reasonable probabilities for the true number of contributors, particularly since we're relatively confident that each electropherogram is exhibiting some signal from each of our contributors. And so the threshold that we chose here was 0.001. Now, I want to be very careful here, and I want to I want to be clear that this 0.001 is not necessarily a recommendation that we have, but it was something, a starting off point for us, because we wanted to make sure that, again, the output was reasonable for our samples. And so what I'm plotting here is the accuracy, or the number of times out of 815 for condition 1, for run 1 of condition 1 and condition 2 and condition 3, basically the percentage of time the probability for the true number of contributors exceeded 0.001. And we see that across all of our sample complexities, the numbers, especially between condition 1 and condition 2, are quite high. Condition 3 starts to fall off, not only in terms of precision, but again, in terms of accuracy. So again, as I said, we chose condition 1 to move forward. So next is our stacked plots, which I found very helpful to plot and to use as I was trying to migrate through all of the data and try to understand the output of NOCIt across all of these 815 samples. So again, knowing that we chose, this is representative of condition 1 results for run 1. And the way to read this is the y-axis represents the probability for that number of contributors for n is equal to 0, 1, 2, 3, 4, 5, 6. And so here the orange bar, if we look at sample 19, for example, we know that the true number of contributors here is 1. And what this is telling me is that sample 19 nearly, NOCIt's APP at n equals 1 was nearly 100%. But here for sample, what does this look like? Sample 85, what this is telling me is that I didn't have just one prominent bar suggesting that, one number of contributors explains the evidence well. What I had was probably two bars wherein the n equals 1, the probability of n equals 1 given the evidence was around 0.9, and then the probability that n equals 2 given the evidence was n equals 2. And so these are important visual tools because it'll tell us, at least for the samples that we tested, that it'll give us an overall idea of how many times we're quote unquote underestimating and overestimating given the true number of contributors. And I do want to say, from a research perspective, I think it's very difficult to compare paper to paper, the number of contributors, accuracies and results, and it's something I think that needs to be discussed a little bit within the community, simply because the accuracy for any number of contributor software is going to be highly dependent on the types of samples chosen to perform the accuracy tests. So again, this is one of the reasons why we did choose to use the proved-it samples, because then this way we were hoping that the community would be able to compare sample to sample across the multiple number of contributors platforms, rather than having to just try to guess whether or not the groups were, quote unquote, fair in choosing the complexities of their samples, and trying to decide whether or not the groups or the developers were, justly, used up to, X number of contributors, or like, did they make the samples complex enough, and all of that stuff. So we thought that from a research perspective was really important. It's something that I do want to continue to shed light on, that using the proved-it data set to us was an important part of this validation and the research. Nonetheless, so here what we have in the pie chart is the percentage of samples across the 815 wherein we only obtain one number of contributors with probabilities greater than 0.001. And here we see that 54% of our samples, we got two probabilities greater than 0.001. And similarly here, we only got 12% of our samples with probabilities greater than 0.001. So this is quite significant, both from a validation perspective, but also from a research perspective, because what it's suggesting to us is that for the most part, at least from NOCIt's perspective, most of the samples are well explained with a number of contributors of 1 or 2. So that is wherein we could choose 1 or 2 values for the number of contributors, and we could be relatively confident that a great proportion of the probability range is being included. So how does that impact casework? Well, it impacts casework because these data, this research suggests that it's, at least for these samples, that we didn't see many cases or any cases, for example, where either 1, 2, 3, or 4 people explain the evidence well, right? We only saw in some rare cases, or 12% cases, where a range of 3 numbers explain the evidence. Most of them were explained by 2. The other thing I do want to point out that we found important was that our APP distribution is always unimodal. So what does that mean? It means that we didn't observe an instance where the probability for a low number of contributors was really high, and then it dipped down for a higher number and then went back up. So you can envision, it's not as if we ever saw an APP of 0.8 at n equals 1, and then it dips down to, say, 0.01 for n equals 2, but then goes back up to 0.2 for n equals 3. Basically, what we always saw was in our probability distribution, the ranges were continuous, right? It was between 2 and 3, or 2 and 4, or 1 and 3. It was never 1 and then skip 2 and then 3 and 4. And that's something I don't think gets a lot of discussion, but when we were validating NOCIt's APP, that's something that became an important part of our validation, because of course our expectation was that we would have unimodal distributions. Okay. So this is a very busy slide. I do understand that, but we're gonna cut into it one slice at a time. So, but this is an important slide for us, and it's presented in this way in the paper because it tries to explicate or tries to explain or delve into all of the questions that at least we've obtained from either other scientists or practitioners, or indeed the reviewers of the paper, which we thought were all very legitimate and helpful questions. So the way to read the chart, I'll focus on this first chart just so we can determine what I'm trying to show you. So again, recall, if this is the the probability of n given the evidence, in this example, this is the NOCIt output. So in this example, the probability that n equals 2 given the evidence is quite high. It's about 0.8. And again, for the for 3 people, the probability is about, looks like 0.2. So as I mentioned to you before, the accuracy is going to be, if I classify something as accurate, it's going to be highly dependent on whether or not whether or not the probability exceeds some threshold that I, as the scientist, have implemented. So one of the things we were interested in examining was, well, what is the effect of my APP threshold? So for this example, if my alpha threshold is 0.5, that is, I'm not going to include, say in this example, n equals 3 as a possibility because it doesn't reach a probability of 0.5, then what this does is it forces us to only use the maximum, the value associated with the maximum probability. Now, however, if I was more interested in focusing on reasonable numbers of contributors to exclude rather than trying to focus on choosing the most probable number of contributors, then I might relax my alpha value, my alpha threshold, and say, okay, well, as long as my probability exceeds this threshold, I'll count it as accurate. So if this is a true 3-person contributor mixture, if my alpha value was 0.001, then I can see that my probability for 3 exceeds this value, so I would have classified this as correct or accurate. However, if my alpha value was 0.5, then in this example, because my true number is 3, I would have classified this as incorrect. So let's take a closer look at the bar chart. So this represents the proportion of samples wherein the APP for the true number of, in this case 1, is greater than my alpha. And here on my x-axis are my alpha values. Now these again are arbitrarily chosen for this paper, but we believe we established a relatively good range. And notably what we see is that the accuracy, of course, increases as I relax my alpha criterion, if you will, but it doesn't actually dip that much. So 84% of my 101-person samples came back with the maximum probability for n equals 1 being at least 0.5. And wherein 90% of my samples gave me an alpha or an APP of at least 0.001. So that's how to read this graph. And interestingly, I do want to point out that the solid bar across the samples represents my accuracy when I used MAC. So for the MAC method, we used a threshold of 100. RFU for the analytical threshold, and for the stutter filters, we just use the global filer stutter filters that are published in the manual, the manufacturer's recommendations. And you'll see here that the accuracies are really quite low. At least for us, it was surprisingly low for the single source samples, and it was at 66%, I believe, or 0.66. And when we went back and looked at why our MAC accuracies were as low as they were, it became clear to us it's because when we randomly chose the 100 samples from the proved it data set that we were going to test, we just happened to randomly choose very degraded and UV damaged samples. So in many of these instances, we had higher stutter values or stutter percent or stutter peaks that would exceed our typical threshold. So it does bring to light this notion of, whether or not we can categorize samples as simple versus complex. I think this is a good example of how even single source samples have the potential to be relatively complex. But nonetheless, we see that there's, depending on the threshold we choose, there's some effect on NOCIt's accuracy, but not not too much. And then if we focus on our true number of contributors 2 and 3, we see that if our alpha, our threshold is 0.001, we have very high accuracy rates. That is 97% of our 2,000 samples NOCIt categorized it correctly. It's suggesting that 2 persons explain these evidence quite well enough. And then it's also for the 3-person samples and 4 and 5. And clearly what we see is that as we move into, in particular, the 5 contributor samples, if we rely wholly on what I'll say is a pretty stringent alpha value, then our accuracies do dip down quite a bit, and notably probably because there's a little overlap and things of that nature that make it a little bit harder to assess to determine whether or not 4 or 5 persons contributed to the sample. And notably here, I do want to point out that for the true number of contributors is equal to 5, the MAC results were 0. So the accuracy of MAC was 0, and this dash line represents the accuracy associated with an MLE method published, so maximum likelihood estimation method published by Hannett and co-workers, I think back in 2011, where they didn't look at peak heights, they didn't model peak heights per se, but they did look at modeling the frequencies, and it does perform better than MAC, and notably NOCIt obviously performs well here as well. So that's how to read the bar charts, and these are bubble charts representing on the y-axis the MAC assignment versus the probability for the true number of contributors, and the size of the bubbles represent the percentage of samples that fall within the category. So this is the single samples, and we see that out of the 66% of the samples that came back wherein MAC suggested 1 contributor, in many instances NOCIt also suggested, gave a high probability for n equals 1. But the interesting parts of this graph are actually those bubbles that are colored in a darker shade, because that represents the number or the percentage of samples that MAC categorized as 2, but NOCIt categorized as 1. And similarly here, these are the percentage of samples wherein MAC quote unquote got it right, but NOCIt's APP was close to 0. So in the top right here, we see the number of times where NOCIt had an APP greater than 0.5, but MAC classified the sample with an incorrect number of contributors, and the number 6 representative of the reverse occurring, where MAC got it right and NOCIt if you got it wrong. And in these graphs, we're using an alpha of 0.5, right, rather than 0.001. And the more interesting parts of the graph, of course, are here at the higher true number of contributors levels, where we see there are often many times where NOCIt is helping us distinguish the higher number of contributors, and MAC is not doing as good of a job, and neither is the MLE method, which is also reliant on filters and thresholds. So the other thing we did is we looked at the probability of correctly classifying, or I don't like to say this, correctly including the true number of contributors into downstream interpretation, and we modeled that against the degree of electropharogram sloping, which I will remind you is modeled using this equation here, and is also graphically depicted using this gray line. So if there's quite a bit of degradation, then this mu value will be very negative, and if there's not a lot of degradation, then that value will be close to 0. So if we look at, for example, these plots, which are split up amongst the true number of contributors and also split up between like very low amounts of very low amounts of minor contributor all the way up to not, major contributor, we can see that the variable or the feature having the highest effect on accuracy is indeed degradation, which makes sense because if we're missing half of the profile, then it's going to be harder to determine the true number of it's going to be harder to determine the number of contributors accurately and include that number in downstream interpretation. But one thing I do want to point out as well is because of the samples that we were testing, the number of contributors, if there's no degradation, our accuracies, NOCIt accuracies are quite good. And indeed, they remain quite high, even up to relatively high degrees of sloping. So again, the sloping of negative 0.02 is quite dramatic, as you see here. So just to reiterate, as expected, using less stringent alphas, or you can think about it as assigning multiple numbers for downstream interpretation, it always improved the chances of correctly including the true number of contributors. And all impact variables affected the chance of including the true number of contributors. But interestingly, it did not do so unidirectionally. And again, that has a lot to do with the type of single source samples that we were testing, and that the great majority of them were they were UV damaged and quite degraded. So the other thing I do want to point out is that, again, NOCIt doesn't require an analytical threshold. One can apply an analytical threshold. It doesn't require one not to, but you don't have to apply an analytical threshold. So it was of interest to us to determine whether or not there would be some effect at the boundary. And so what we did is we ran a negative amplification control where we didn't see any peak that crosses the analytical threshold. And so if you essentially run a blank sample in NOCIt, what you get back is like not that much information or no updated information. And so what this is telling us is, well, if you have no data, then the number of contributors that may have contributed to that sample with no data is even across the board, right? We can't say that much about, we can't really say anything about that sample. But if we ran with no analytical threshold, then that NOCIt returns a probability of n is equal to 0, which is essentially 1, very close to 1. And so was an interesting result for us because it did suggest that there was some information that could be used, which is typically considered, is typically considered maybe information that has not a lot of value, but for the number of contributors aspect of things, we would argue that there is some value in those peaks that are below these, these high-ish analytical thresholds. So in conclusion, we did conclude that in our view, NOCIt's APPs performed relatively well, and indeed really well, and they were quite robust even across the most degraded and damaged and inhibited samples. I do want to point out that of the 815 samples that we did test, I think 45 of them or 50 of them were also differentially degraded. So we did try to produce samples that represented all of the complexities that typical casework laboratories have to deal with. And I do want to point out that to our mind, all samples are complex, and even single source samples. So this is quite obvious in the single source results, at least from the number of contributors perspective. And the greater than 1n is needed if inclusion of the true number of contributors in stream interpretation is the aim. And again, that depends on whether or not it's the aim, and it again would be quite dependent on the upstream laboratory conditions as well. And in the end, no analytical threshold is required to complete probabilistic interpretation. It's a choice that is made, but it doesn't have to be made in that way. And indeed, part of NOCIt is that we don't require it, although it can be applied. So real quick, I get a lot of questions about, well, how can I use the APP? Well, it would depend quite a bit on the reporting practices of the laboratory, but in essence, there's a number of ways, and I discussed two ways previously. One is to just pick the number of contributors affiliated with the highest probability. So you're choosing probable number of contributors given the evidence, and that's your assignment, and you compute, or one computes one likelihood ratio, and that's what's reported. The other method is to exclude unlikely number of contributor values from downstream interpretation. And then yet a third method is to the full likelihood ratio, where the probability of the number of contributors given the evidence is what's known as the nuisance variable. And I do want to point out that this is not work that, was explained by us. This is a research paper, a very nice research paper from Slutin and co-workers. So I would go ahead and encourage everyone to read that. But at the end of the day, any number of contributors estimation has to be integrated somehow into the pipeline. And so in general, the pipeline includes the generation of the data and then the analysis of the data. And the analysis of the data, of course, can be quite complex because it has its own intricacies related to artifact filtering, and is this a peak, is this not a peak, what should my analytical threshold be, etc., etc. So we in our laboratory sort of, I think, understood the complexities associated with the analysis process. And so as part of the NOCIt pipeline, if you will, we did add a cleaning module so that manual filtering of artifacts doesn't have to be done by the analyst, though of course it can be supported by the analyst, and that would be up to the laboratory. And of course the reason we put that in, CleanIt, we call it CleanIt. The reason we put CleanIt in there is because we recognize that with lower analytical thresholds, there's going to be more artifact labeling, and so we did want to provide any user the ability to filter artifacts, as well as but not including stutter. So the -A artifacts and especially the pull-up artifacts. So that's part of the module. That's a module of NOCIt, though I won't be talking about it too much today. And then we have the model parameterization that needs to occur, and that is done through a module called CallIt. And then there's the number of contributors evaluation. That's NOCIt. That's what I validate or showed or demonstrated the validation of today. And then there's CSIT, which is the platform that computes the likelihood ratio. Now, CSIT and NOCIt use the same underlying probabilistic models, which is a condition for using this equation. So what that means is I could take the NOCIt result, the APP, results and multiply it by the likelihood ratio obtained for that n, and then sum across all the non-zero values of APP. So we found that one of the interesting things about NOCIt is that because it allows us to look at the probability distribution ahead of time before we actually run all of the likelihood ratios, we could exclude basically negligible values for n before we go, before we calculated the full number of contributors, or excuse me, the full likelihood ratio. So let me take a step back. So this is simply an example of how one would go about doing it. So again, going back to our original sample or example, which is, if you recall, NOCIt suggested there was a non-negligible probability that n equals 3 explains the evidence. Then we could take the APP obtained from NOCIt and multiply it by the likelihood ratio from CZIT for n equals 2, and then add it to the APP for n equals 3 by the likelihood ratio when n is equal to 3. And so this would be the overall or the full likelihood ratio without having to actually choose a number of contributors or a single number of contributors to acquire that value. So I do want to share with you that NOCIt is available for download. So there's a license, an academic and research license, 6 months license for any lab or research lab that wants to evaluate it or conduct a research experiment or do a study with it. And the way to get to it is to go to lifty.com. And again, just to remind you, the software package is not called NOCIt, it's called ValidDNA. NOCIt is simply a module in that software program. And all you have to do is hit obtain a copy, and it brings you to Rutgers, commercialization office, and you fill out a form and it downloads for you. So I just want to thank again my long run collaborators, Desmond and Ken Duffy, and I in particular do want to thank Slim Karkar, who developed the biomodels or the analytical models, and Lauren and Amanda, who generated all of the proved data. And Zia, I didn't talk too much about it, but Zia Yearwood Garcia, who's a student at Boston University of mine, she did all or a great majority of the software testing for the GUI, which is if anyone's done software testing, it's not the funnest thing to do, but she was great at it. So I just want to give her a shout out there. So now I just want to go ahead and quickly demonstrate to you what NOCIt or what ValidDNA looks like. I understand it's already 2:10. So real quick, here's the call it module here where the models are, where the samples are, let me try that again, where the calibration file imported and the models are therefore parameterized. So I'm just going to go ahead and open a calibration project that we've already created just so you could see what this looks like. So this is something that only has to be done once for the laboratory for every laboratory, process or condition that they implement. So we see here that, so these are the samples of known genotype. They're all single source samples. And what you have here is a sample name. And we're designating whether or not we want these samples filtered by CleanIt. And we also affiliating the known genotype of 39 with this sample, which of course, as I know based on this name, is from person 39. And so by affiliating the known genotype with said sample, we can then develop the various models that we implement. Everything is done on a per locus basis, and there's this graphical representation of in this case the peak height with respect to this this feature that we call decayed amplitude. And one of the things I always tell new users to do is when they're first trying to parameterize the models, always go to the noise peaks, because you could see, you'll know if you did it correctly, because your noise peaks should always be around, depending on your instrument, should always be less than about 100 or so. Of course, there are going to be a few instances, i.e. drop-in instances, or the fact that one didn't, where CleanIt didn't catch it, or maybe the analyst didn't catch it, or a spike that got through the system. You might see something that's classified as noise, but it has a really high peak height. So this is a good way to QC your calibration of your samples. And so once the models are created, simply save this, what we call calibration project, and this project is used throughout all of the evidentiary evaluations. So you can say that, okay, if you're interested, for example, in running some evidence sample, one would have to load the calibration project that one created for that injection time or that cycle number. Then we would browse to the output folder of interest. So you just browse to whatever location, either on the computer or on a network. And also, if you wanted a summary file, not just a PDF file of all of the results from NOCIt, you could go ahead and ask to create that CSV file. And then here you could choose the maximum number of contributors for which you want to test. So if, again, one of the parameters that, or one of the things we could choose, or a laboratory could say is, well, I don't interpret anything above 4 contributors. If there, or I don't want to anyway, right? It's hard to know if something comes from 4 contributors, but let's just say that's sort of a criteria that the lab has in mind. Then there's no need really to test up to compute the APP for up to 6 contributors. You can perhaps choose 5, because anything that would be above 4, or anything that would be 4 or 5, you would know it's too complex for your laboratory protocol. And so by decreasing this number, and you would decrease the runtime as well. So to add the samples, you could add any samples in a batch, and the samples are just CSV files, just like this. So it's exactly what would be exported from any gene mapper, gene marker, or OSIRIS file. I believe most of you are used to seeing the data in the wherein we have the sample names on the first column, then the marker, the dye, and then all of the information, allele size and height, allele size and height. So it does need to be ordered in this way. I mean, NOCIt's always going to be looking for the allele size, height, triple, if you will, in that order, but that's something that you have control over. And so here you could decide which samples out of all of your samples you would like to run by simply Xing out those you are not interested in running. And if you did apply an analytical threshold during analysis, this is where you would tell us you did so. So if you use an AT of 50, you would tell us that you used an AT of 50 for your data here. And here's where you would choose your populations, the frequency file, and these are just the typical, this is just the CSV file wherein we have the allele designator and then the frequency, well, these are percentages, the percentages of those alleles observed in that population. Now the important thing, well, I'll just show you. So you can add the entire batch, and then one of the nice things is the table is quite versatile. So if you don't want to run the entire 815 samples, which I'm sure we do not, you can go ahead and just leave the one sample that would be of interest to you. And then if there's some sort of case, you know, file or number associated with it, you can go ahead and choose all of that. The other thing that is of that's interesting, at least to us, is that you can also choose a, you know, a known contributor. You know, so if there's a victim or from a sexual assault, and you're interested in knowing the probability for the number of contributors, you know, given there's an unknown plus a tethered victim or a known contributor, you could go ahead and choose, you know, multiple known contributors and then run NOCIt in that way. So the output files, I'll just quickly show you what the output files look like. They are PDFs, and it tells you the analytical thresholds you used, and it tells you the probability for that number of contributors given the evidence. And then on the bottom, there's a graph. And similarly, you can choose to export all your results as a CSV as well. So you don't have to always be opening up PDFs. You can go ahead and open up, you know, an entire batch's results by choosing to export the CSV. And the only last quick thing I'm going to mention is that NOCIt is completely, I mean, the loci that one uses in order to let me try that again. So the loci and the kits are completely dependent on what we call as the bins file or the kits file defined by the user. So this is important, and essentially it's just the bins file. It looks almost exactly the same as the bins file from GeneMapper, you know, that's used in GeneMapper or GeneMarker. And this is important because if, at least to us it is, because we don't always use the same kit, and we wanted some flexibility with respect to being able to define the loci and the alleles that we wanted to use. So it's not hard coded in valid DNA. So the mark, say you only wanted to test D3, then one would have to do is get rid of all of the remaining bins and loci that are no longer of interest to you. And then what we would see is that only the D3 locus would be modeled, and only the D3 locus would be the probability for that number of contributors given the evidence would only be based on the D3 locus. And so this bins file, even though it's relatively seemingly innocuous, it's actually one of the most important parts of NOCIt because it is the thing that defines the loci and the alleles, and it's completely controlled by the user. So that's the tutorial there. And since it's 2:20, I'm gonna invite Josh back and see if there's any questions that I can answer. And I just want to thank everyone for giving me your time today.
All right, thank you so much, Catherine. Before we jump into the questions that have come in, I would make sure that everyone knows that we have recorded today's webinar, and we will be converting that into an archival version over the next few days. There was a lot of information presented today, so we want to make sure that you do have access to that and can come back and reference it or pass along to any colleagues that might have missed today's live event. Also, as Catherine mentioned, this was a postponement from last week, but we are pleased to acknowledge that we did have well over 200 people to come back and view today's event. So thank you everyone for making the time in your schedule to come back for today's webinar. Also, Catherine has provided a PDF version of her slide deck. It is back on the main webinar page where you first came into today's event. So do please make sure you download that before you leave today. So Catherine, there was a question from one of our international users. It was speaking directly about his country, India, but I'm sure this could go for all international forensic users as well. Is it possible to get the NOCIt in India for FSLs?
Yes. Yes, you just go, yes. So you just go to the website that I showed you before, you click obtain a copy, you fill out the license, the information, and then there's an automatic 6-month user license that will be downloaded onto your computer, and along with that license, valid DNA will be downloaded. I want to point out that, I mean, it would be necessary to read the license quite carefully because it is a research and academic license. So to acquire a license for casework, it's our view that there's a significant amount of training that would still need to be provided to the user. Because of course, like for example, today I skipped over the models in their entirety almost, right? So that's something that we would provide should the user, evaluate NOCIt, think it's something they wanted to use in casework, and then there would be a distinct casework or commercial license that would be required.
All right.
For students and professors and things like that, or labs who want to evaluate NOCIt for their own validation and research purposes, that's the way to get it. And the download happens within a few seconds.
Fantastic. We do have quite a few just thank you and comments. Many people were saying that this was very informative. So you definitely did a fantastic job explaining everything. Since it does seem like this program is so robust and you did mention the training, would you mind going over a little bit of that once they do get approved? I know that would be a vital part to anyone that does fill out the application and want the NOCIt program.
Well, if they want it, so if they filled out the requisite information, they would automatically get a 6-month user license, and they could use NOCIt for whatever purpose they wanted, right? So it could be that they wanted to evaluate it against, maybe they're not using a 3500, or maybe they're using a 3500 XL, or they have very particular samples that they want to make sure NOCIt is not just working in our hands, but that NOCIt is working in their hands. This way they don't have to contact us or pay for anything. They could just go ahead and evaluate NOCIt and see if it's something that they're interested in using for casework. It's at that decision point where they would then, if they decided that NOCIt was of interest to them, then they would just contact me, and then I would provide them, we'd set up a time and a schedule and some sort of pay scale, or we would organize the full training. The training usually takes, the initial training is probably anywhere between 1.5 and 2 days, and it would be highly dependent on the needs of the laboratory. So that's basically done on a case-by-case basis.
All right, fantastic. We do not have any other questions that have come in, and since we are a little bit over time, we are going to go ahead and wrap things up. But oh, actually, we do have a few that just came in. So real quick, so for casework after 6 months user license, can it be renewed with the same procedure, or what would be the next procedure after that?
For casework, so let me make sure I understood the question. So if 6 months was not enough time for evaluation or for research, then yes, you could just use the same process and get the license for the research license for an additional 6 months. If, however, after 6 months, the crime lab or the tech lead or whoever it is says, we're really interested in implementing this, we want to make sure we're implementing it correctly, then they would just contact me, and then I would get them in contact with the Office of Commercialization so that we could develop a full license, a full casework license, and then we would go ahead and schedule the training. So you can, and then you can contact me using the email below that I've been showing, which is C. or you can contact me through the Lifty website. Either way is perfectly fine.
All right, and definitely make sure that you download a version of today's slide deck. That way you do have Catherine's email address there, as well as the websites and resources that she provided. With that, we are going to go ahead and close up. Oh, actually, we had one more pop in. The person that asked about NOCIt in India is saying that they need it for the forensic science laboratory for regular casework.
Mm-hmm. Then you're more than welcome to contact me, and I will work with you to get you a copy. Just email me at c.gergachuk@Rutgers.
Fantastic. All right, with that, we are going to go ahead and close up. Obviously, we would like to take this time to thank Catherine, Dr. Gergachuk, for taking the time out of her day again to present this material to us. So thank you so much, Dr. Gergachuk. We definitely appreciate you coming back today and giving your presentation.
Sure. You're welcome. Thank you.
We would also like to take this time again to thank our attendees for joining us today from all over the globe. These webinars are simply not possible without your attendance and your support. Before you leave, I ask that you participate in our short survey. You will find that directly below the closed captions on the right side. Simply click the word survey, and that will pop up a new window for you where you can let us know your feedback about today's webinar and how it was delivered. We always take that information very seriously, and we want to make sure that we are always providing you the best experience possible for our webinars, as well as adjust any content or the way that we disseminate that information out to you, our audience. So if you could take a few moments to download that before you leave, we would definitely appreciate it. You will see in a few moments, Tyler will be sending links through the chat pod for our social media outlets, LinkedIn, Facebook, and Twitter. Also our website, where you will find all of our no-cost webinars, reports, success stories that the FTCOE is constantly putting out to you, the forensic community. You will also see a link to our podcast. So do make sure that you're following us on all of our outlets. Again, we did record today's webinar so that you will be able to access that and refer back to today's material. And again, make sure that you do download a PDF version of today's slide deck. Again, we hope everyone attending today found today's webinar very useful and informative. We are constantly adding new content to our website, and we hope to see you next webinar that we host next week, and we hope you all have a great rest of your day. Thank you for joining us.